Hi there, my name is Mandeep Mann, and I am a scientist in training at the University of Toronto. Developing a new drug against ever-evolving diseases and conditions is a long and difficult journey. It takes years and millions of dollars. Genome sequencing and genome editing efforts have identified potential drug targets for the possibility of inventing new medicines. These drug targets are proteins. Proteins are the machinery in the body. They do all the tasks that we don't often think about, things like breaking down food or providing structural support in our bones, skin, and hair. My research focuses on one of these proteins called USP5. USP5 is a protein that is involved in the balancing act of protein degradation in the body and has been shown to be essential in most cancers and is also associated with chronic pain. The first step in understanding USP5 and the role it plays in cancer and chronic pain is to develop tools to be able to study this protein. What is its function? How does it work in normal and healthy cells? And what is the role of this protein in the disease? To do this, my research goal is to develop a chemical probe, a drug-like molecule that will bind tightly and specifically to USP5 in cells. To accomplish this, I use computational chemistry to predict, design, and optimize small molecules that bind to the protein. I then test these compounds in the lab. Do they bind? If so, how well do they bind? Do they bind in the same way that was predicted? Once these questions are answered, I can go back and make these compounds better. The end result will be a chemical probe, a drug-like molecule that binds tightly and selectively to USP5 in cells. This chemical probe can then be used as a tool by me and other researchers to study the biological role of USP5 in cancer and chronic pain. This tool will be a resource to help accelerate research and really understand what the USP5 protein is doing in disease, and possibly discover novel medicines more quickly.